Welcome to the AP Biology review of plasma membranes.
First, we will introduce the concepts of tonicity and osmosis. Then, we will describe the similarities and differences between plant and animal cell structures. We will end by giving a real-life example of cell communication through an example of signal transduction.
Tonicity is the relative property of a solution that describes the osmotic pressure forces present, and it describes how stuff wants to flow from high to low concentrations. A presence of such a concentration gradient drives the flow of material across membranes. A cell is an example of a semi-permeable membrane, which allows only certain molecules across, such as water, through protein channels called aquaporins. Osmosis is defined as the spontaneous net movement of solvent molecules, water being an important example, through a semi-permeable membrane into a region of higher solute concentration in the direction that tends to equalize the solute concentrations on the two sites. For example, say you have a cell whose cytosol has a 0.2 molar salt content, and that cell is immersed in a beaker that contains a 0.4 molar salt content. The salt concentration outside the cell is higher, so relatively speaking, there is more water in the cell than out, so the water flows out of the cell. The beaker solution is called a hypertonic solution. In an isotonic solution, water molecules move into the cell and out of the cell at the same rate, so no difference is observed. This occurs when the concentration of solute is the same in and out. In a hypotonic beaker solution, the cell solute concentration is higher, so water moves into the cell.
As water enters a cell, turgor pressure builds up inside the cell. The free energy present due to more water present inside the cell causes this outward pressure buildup. So here we have a simple diagram of a plant cell. As you can see, the presence of a central vacuole is essential to the cell as it stores water that enters the cell via osmosis and is essential to the turgor pressure of the cell. Also, with a rigid cell wall made of cellulose, the plant cell can handle this turgor pressure, unlike animal cells, which have certain consequences due to hypertonic and hypotonic solutions.
Animal cells, on the other hand, either expand or shrink in response to hypotonic and hypertonic solutions because there is no rigid cell wall to maintain structure. For instance, say we have a solution of 0.4 molarity on the outside of the cell and a 0.2 molarity solution on the inside of the cell. As we have learned, water should flow from the inside of the cell to the outside because the outside of the cell is hypertonic and has the higher concentration of solute. Because water flows out of the cell will shrink.
On the contrary, if the solute concentration is 0.2 molar outside the cell and 0.4 molar inside of it, the outside of the cell is hypotonic and water will flow into the cell. A good example of this is red blood cells. If you have a sample of red blood cells and add enough water to the sample and come back 20 minutes later, you will see that the cells have disappeared. This is because the water has entered the membrane of the cells and the cells have burst as a result.
Now that we have an idea of tonicity and osmosis, we can dive into how stuff flows across the plasma membrane. Since we mentioned the plasma membrane is semi-permeable, the plasma membrane consists of phospholipids. Phospholipids are made of a polar head attached to two nonpolar tails. The polar portion is hydrophilic, which means it likes being in water, while the nonpolar portion is hydrophobic, which means it does not like being in water. Since cytosol and the extracellular fluid are mainly water, the nonpolar tails tend to bunch together in the middle by creating a bilayer as shown. As the center of this membrane is made of nonpolar tails, the plasma membrane tends to let small and nonpolar molecules pass. However, polar molecules and larger molecules must rely on other ways.
Now we will explain the fluid mosaic model. We have learned about the phospholipid bilayer and the presence of a hydrophilic and hydrophobic side. However, there is also a variety of proteins that exist in the membrane, including transmembrane proteins and integral monotopic proteins. These include glycoproteins, which stabilize the structure of the membrane by forming hydrogen bonds with the surrounding water molecules, and cholesterol, which keeps the membrane consistent in terms of fluidity.
Now that we've learned what the cell membrane looks like, let's talk about how things move inside and outside of the cell. As Anish said a bit ago, small and nonpolar molecules, like some kinds of lipids, can pass right through the semi-permeable membrane. This is simple diffusion. Big and/or polar molecules need different ways though. When a non-diffusing molecule is transported along its concentration gradient, or from a higher to lower concentration, it can travel through specialized proteins in the membrane. This is passive transport, as even though the molecule can't just diffuse over the membrane, it doesn't need the investment of additional energy. In the case that a molecule is moving against its concentration gradient, like certain processes in neural signal transduction, it needs additional energy. The molecule will still travel through specialized transport proteins, but these proteins will need ATP to work. Finally, the transport of water requires its own proteins. Even though water moves along its concentration gradient, reliant on tonicity, it can't flow through the membrane alone due to its polarity. It thus moves through aquaporins, a special type of facilitated diffusion protein.
Let's end by applying what we've learned to a real-life example. Epinephrine is a hormone typically produced in the adrenal glands. It attaches to an epinephrine receptor protein embedded in the membrane of a cell, and thus leads to an involved process of cellular communication called signal transduction, and ultimately leads to the breakdown of glycogen into its individual glucose monomers, which would then be used for cellular respiration. When epinephrine binds to the protein, G protein is activated, binding to the receptor, taking up a molecule of GTP and releasing GDP. After consuming GTP, the G protein is released and broken into its subunits. One of those subunits activates an enzyme also embedded in the plasma membrane called adenylyl cyclase. Adenylyl cyclases convert many ATP molecules into CAMP molecules, which are referred to as the second messengers within the cell. CAMP molecules then bind to protein kinase A, taking the regulatory subunits off and releasing the catalytic subunits, which add a phosphate group or phosphorylates an enzyme called phosphorylase kinase, which goes on to phosphorylate glycogen phosphorylase, which as a result is activated to break down glycogen into glucose. Good luck on the AP exam. Bye.